Imagine walking through a forest with the sound of birds, the smell of damp earth, and suddenly holding in your hands a fragment of the past with tens of millions of years. These creatures did not come out of a science fiction movie. They are real, and they were found perfectly preserved in amber, the fossilized resin of prehistoric trees that captured moments of life on the planet up to 200 million years ago. Today you are going to dive with us into a lost world where time has stopped. The oldest arthropods ever preserved in amber. $230 million years ago, when dinosaurs were still taking their first steps on Earth, a silent and almost imperceptible event forever sealed a primordial chapter of microscopic life. In the vast tropical forests that covered what we now know as the Dolomite Alps in northeastern Italy, a tree released its thick, golden resin, dripping slowly and continuously. That resin, which for moments shimmered under the warm Triassic sun, captured, as if in a fossilized enchantment, two of the oldest arthropods ever found in amber. They were mites, tiny creatures, almost invisible to the naked eye, yet carrying in their biology the genetic story of eras long gone. Preserved with an almost poetic perfection, they have rested for hundreds of millions of years in amber capsules the size of rice grains, measuring between about 0.08 inches and 0.24 inches in length. Invisible to the untrained eye, yet revealing under the careful observation of modern science, these traces are not alone. They were discovered by an international team of researchers, led by Eugenio Ragazzi and Guido Roghi from the University of Padua, and Alexander Schmidt from the University of Göttingen. Modern chicken mites are known for their specialized diet. They feed almost exclusively on flowering plants. But the specimens preserved in this amber belong to the Triassic period, an era when flowering plants did not even exist. How then could this creature have survived, thrived, and adapted to an environment so different from the one we know today? The answer lies in their resilience, in their ability to adapt to changing worlds. Before flowers, there were ferns, cycads, and conifers, vegetation that dominated the Triassic landscape. And it is likely that these plants, with their nutrient-rich tissues and primitive pollen, served as the first feast for these ancient mites. Their bodies suggest a slow and methodical locomotion, ideal for dense plant environments. The sensory hairs indicate tactile and perhaps even chemical sensitivity, fundamental for finding food or reproductive partners in a forest where competition was constant. Bird hatchling, Belone. In a piece of Burmese amber, covered by a patina of centuries and wrapped in layers of sediment and silence, rests a creature that seems to have come out of an interrupted dream. A hatchling. Fragile, light as the breath of an ancient memory, frozen in time 99 million years ago. This is Belone, the most complete bird ever found in amber. Imagine finding not just a feather or a bone, but an almost entire fragment of a winged being that lived among dinosaurs. Half of a body, part of a wing, a hind limb. The skull with its expression erased, and even the delicate curve of the tail still wrapped in soft tissue. Belone is not just a fossil, it is a visible ghost, a natural reliquary that preserves the exact instant when life and death touched. Belonging to the group of Enantiornithians, Belone is part of a lineage of primitive birds that coexisted with the great reptiles and disappeared with them in the cataclysm that marked the end of the Cretaceous 65 million years ago. But while most of these fossilized birds reach us as flattened impressions in rock, below came wrapped in resin. Intact keratin sheaths surround the toes, revealing that even in the end it still clung to what it knew. The branch of a tree, perhaps, or the air itself. The smallest bird dinosaur in the world found in amber. In the heart of the dense forests of northern Myanmar, a treasure of time was discovered, trapped in golden amber for almost 100 million years. Inside it rested the incredibly preserved skull of the smallest dinosaur known to this day. Its name, Oculudentavis kaungrai, sounds as if it were taken from the pages of a science fiction novel, but its existence is real and extraordinary. The resin that encased it about 99 million years ago preserved anatomical details that would never have survived the passage of time under normal conditions. Its skull, measuring only about 0.55 inches in length, revealed to researchers a true enigma of nature. It was not only its size that surprised them, it was its morphology, its set of adaptations, and above all, its hybrid identity, part bird, part reptile. The creature's name, translated as bird with teeth in its eyes, refers to the most striking anatomical features of the fossil. Oculudentavis possessed around 100 sharp teeth, small serrated cones like crystal blades. This impressive dentition, arranged along a thin and elongated jaw, suggests that the animal was an agile hunter of small invertebrates, possibly capturing its prey with quick and precise bites. But what intrigues scientists the most are its eyes. Large and circular, resembling those of certain lizards, the eyes of Oculudentavis had a very narrow opening, a fixed pupil that allowed only a limited amount of light to enter. This indicates that the animal was probably diurnal, active under the intense light of the day, unlike many reptiles and birds with nocturnal habits. Measuring about 2 inches in length from head to tail and weighing approximately 1 ounce, Oculudentavis was even smaller than the smallest modern bird known, the bee hummingbird of Cuba. But unlike its modern relative, this Cretaceous creature did not have a beak adapted for sipping nectar, but rather a predatory dentition. Its body, which scientists were able to digitally reconstruct based on the skull, was probably covered with thin, aerodynamic feathers, similar to those of a primitive bird. Feathered dinosaur tail. In 2016, in the tropical forests of northern Myanmar, a fragment of fossilized resin was extracted from the earth. At first glance, it seemed like just another beautiful piece of Burmese amber, so coveted by collectors and jewelers. But upon closer examination, it revealed something extraordinary. Embedded in the amber was a tail, not from an insect, nor from a small reptile. It was the tail of a dinosaur. A fragment of the past, preserved with almost miraculous fidelity. This rare and magnificent fossil carried with it not only bones but also feathers. Real feathers, thin and delicate, arranged along an articulated tail with visible vertebrae. A discovery that would forever change the way we perceive dinosaurs and their connection to modern birds. We were witnessing a turning point in science, the moment when the myth of the scaly, brutal dinosaur gave way to a new image, that of the feathered dinosaur, light, agile, and surprisingly modern. The tail belonged to a juvenile theropod, a member of the same group of dinosaurs that includes the fierce velociraptor and the imposing Tyrannosaurus rex, but also the direct ancestors of birds. It had brown and white feathers arranged in rhythmic patterns, with structures resembling those of modern bird feathers, although they were not adapted for flight. These feathers served other purposes: thermal insulation, display, camouflage, evolutionary tools as effective as wings. Microscopic analysis of the fossil revealed impressive details. The feathers had rachises, the central shafts that support each row of lateral barbs. These barbs, in turn, were not interconnected by microscopic hooks like the feathers of modern birds, which indicates a more primitive structure yet still fully functional. The tail preserved 8 separate vertebrae, a rarity in fossils, and these vertebrae were not fused, confirming that the animal was still very young. The first baby snake of the dinosaur era. Preserved with an almost unreal delicacy, there lay the slender and fragile body of a baby snake, a creature so small, so vulnerable, that its existence nearly 100 million years ago seems like a whisper lost in time. This is Geophis myanmarensis, the first baby snake of the dinosaur era ever found fossilized in amber. With an estimated age of 99 million years, this fossil represents not only the oldest specimen of a snake in a forest environment, but also the best preserved record of a juvenile snake from the Cretaceous period. Before this discovery, we had never seen with such clarity the skeleton of such a young snake, preserved in 3 dimensions, with dozens of intact vertebrae, visible muscles, and the translucent silhouette of its body still recognizable. Found in an amber deposit in northern Myanmar, the fossil was studied by an international team led by Lida Xing of the China University of Geosciences. The group named the new species Geophis myanmarensis, a name that honors both the geographic location of the find and the tiny size of the creature. Xiao, which means small in Chinese, is a direct reference to the youth of the animal. The baby snake likely lived in a humid, densely forested environment, very similar to the tropical forests we know today. This is particularly important because previous fossil records of snakes generally came from arid regions or riverine environments. Geophis is the first to show us that snakes also inhabited dense forests during the Cretaceous, expanding our understanding of their ecological and geographical diversity. Amber, acting as a natural preservative, preserved anatomical details impossible to find in traditional fossils. Every vertebra, every rib and every curve of Geophis's sinuous body was kept almost intact. Through ultra-high-resolution computed tomography scans, scientists were able to observe the microanatomy of the skeleton. Although the skull was not present in the fossil, the rest of the body provides precious data. Researchers identified more than 100 articulated vertebrae, with a degree of ossification that reveals the animal's youth. The growth pattern of the vertebrae and the length of the ribs suggest that it was still at an early stage of development, perhaps just recently hatched from the egg. Flea with plague bacterium. A flea, nothing more than a dark speck to the inattentive eye, but to science a time capsule that reaches back to a vanished world, a capsule that perhaps holds the ancestral whisper of an invisible threat, the plague. Dominican amber, known for its almost crystalline clarity and yellowish hue, is more than a jewel of geology. It is a reliquary of deep biology. And it was in this amber that Dr. George Poyner Jr. from Oregon State University found something intriguing, and inside its body are perhaps the traces of one of the deadliest microorganisms in human history. Its morphology indicates that even back then it was a vector, a transmitter, capable of leaping between hosts, feeding on the blood of warm-blooded creatures, and carrying microorganisms with it in the process. And one of those microorganisms, which may have resided inside this fossilized body, is Yersinia pestis, the bacterium that much later gave rise to bubonic plague, the same one that haunted medieval Europe, devastated populations, and shaped cities, cultures, and deep fears. But Poynar's flea predates all of this. It lived in a time before humans, before even the idea of civilization, and yet there lay the seed of destruction. Spider attacking a wasp. "100 million years ago, long before any civilization, even before the first primates walked the earth, a silent drama unfolded beneath the damp shadows of the Cretaceous forests. A web-weaving spider, patient and deadly, remained motionless, sensing the vibrations of the world around it. Its web, made of incredibly resistant silk, trembled with the wind and the passing of time. And then, as if by an instant of inevitable fate, a small male parasitic wasp crossed the spider's path, flying straight into its sentence. The wasp never stood a chance. Upon touching the silk, it became trapped. Its movements turned frantic, desperate, but the spider was already in motion. With the precision of an ancient hunter, it approached its victim, beginning a deadly confrontation. This moment, so common in the natural cycle of life, could have been lost forever. A fleeting act, as ephemeral as a raindrop. But something extraordinary happened. A tear of resin dripped from the tree above, engulfing both arthropods in the midst of the struggle. And in that instant, time froze. Preserved in amber for 100 million years, the bodies of the spider and the wasp remained locked in an eternal duel, a predation scene frozen with striking clarity. 15 silk threads, still visible, connect the two protagonists. The spider, an ancient weaver, probably from the family Arcadidae, held its fangs raised in an attacking position. The wasp, on the other hand, was partially entangled in the web, wings spread in a final effort to escape. It is a fossil like few others, an instant of action captured in its purest form. This spectacular record was found in the Hukong Valley, a region rich in fossil amber and mysteries of the dinosaur era. Analyzed by the renowned entomologist and amber specialist Dr. George Poyner Jr., the fossil has become one of the most vivid representations of predator-prey interaction among terrestrial arthropods of the Mesozoic. It is more than a frozen image. The wasp caught in the web belonged to a group of parasitic insects whose habits include laying eggs inside other arthropods. Its role in the ecosystem was vital, helping to control host populations. The spider, with its segmented body and long legs, reveals anatomical traits of primitive social spiders. Lizard from 54 million years ago. Yantaro gecko balticus, one of the oldest and best preserved lizards ever found in amber. With an estimated age of 54 million years, this encapsulated fossil reveals a moment frozen in time. Found deep within Baltic amber, Yantaro gecko balticus is a time capsule, offering a crystal-clear window into the Eocene period, an era when the continents were slowly drifting into their modern positions and mammals were beginning to dominate terrestrial ecosystems. Most reptiles decompose or are destroyed before resin can fully seal them. In the case of Yantaro gecko, fate was generous. The small lizard, measuring about 2 inches from head to tail, was preserved with such remarkable integrity that even its internal organs and the contours of its skin remain visible. The tiny scales, delicately sculpted eyes, and perfectly articulated bones reveal not only the creature's anatomy, but also clues about its behavior, evolution, and role in the ecosystem it once inhabited. The skull, though minute, displays traits very similar to those of lizards in the family Geckonidae, the familiar geckos, which still inhabit various tropical regions of the world. This finding raised a series of questions. Could Yantaro gecko be a direct ancestor of modern geckos? Did it live in treetops, hunting insects among the damp branches of warm, dense forests? Morphological analysis seems to suggest so. Its legs, for example, possessed specialized adhesion structures, very similar to the adhesive pads we see in geckos today. But beyond biology, Yantaro gecko balticus tells an even greater story. A narrative about the planet itself, about the transformation of forests, the resins that turned into amber, the climate and biodiversity. It represents a missing link in an evolutionary puzzle, a silent witness of a prehistoric world. The name Yantaro gecko comes from yantar, the Russian word for amber, and gecko, the generic term for geckos. It is a tribute to the place of origin and its similarity to modern reptiles. Meanwhile, Balticus refers to the Baltic Sea region, where the amber that encased it was found. First salamander in amber. In a very distant time during the early Miocene, a small moist-bodied creature with discrete movements silently roamed the vegetation of ancient Hispaniola. It did not know it was walking through a world in transformation. The Caribbean islands were still shaping their geological identities, and the life that flourished there was a mosaic of evolutionary experiments, fragments of biological stories that, in many cases, would not survive the passage of time. This creature, a salamander, the first ever discovered preserved in amber, lived a little over 20 million years ago. It was found in the Dominican Republic, encapsulated in a natural resin trap that, upon hardening and fossilizing, transformed it into a perfect fossil. A time capsule that survived through ages. Its name, Paleoplethodon hispaniola, an extinct species that, although it left no descendants in the Caribbean islands, tells us today a fascinating story of survival, dispersal, and tragedy. Salamanders are completely absent from the Caribbean today. No modern species roam the moist soils of these islands, none sing beneath the foliage, nor slide between the rotting trunks of the tropical forests. For this reason, the discovery of this tiny creature in Dominican amber was received with both disbelief and excitement by the scientific community. Could it be possible that salamanders once inhabited the Caribbean? What evolutionary paths brought them here? And above all, why did they disappear? The answers begin to emerge in the translucent amber that encased Paleoplethodon. In its fossilized body, a haunting detail leaps out. One of its limbs had been torn off. Researchers, led by Dr. George Poyner, identified clear signs of predation. It had been attacked before being engulfed by the sticky resin. An open wound, perhaps still throbbing with pain, became the harbinger of its sudden death. The violence of a wild world crystallized alongside its image. first ancestors of the chameleon and the gecko. 100 million years ago, trapped in amber and crystallized by the resin of an ancient tree, this ancestral being was kept intact by a quirk of nature, waiting to be rediscovered by modern science. It was in Myanmar, within amber deposits dating back to the Cretaceous period, that this extraordinary creature was found. The small lizard, with its slender legs and elongated tail, had been resting inside a translucent prison since the age of the dinosaurs. And despite the passage of tens of millions of years, it seemed still alive, as if at any moment it could break free from the amber and return to the branches. Scientific analysis revealed something astonishing. This creature was not just another reptile. Its skull and bone structures pointed to a lineage far older than once imagined. It was a missing link, an evolutionary bridge between modern chameleons and geckos. Mixed morphological characteristics indicated that we were facing a primitive stage in the development of these two distinct groups, which today are spread across all tropical continents. Imagine an animal with eyes that could move independently, a projectile tongue, feet with adhesive pads, and an increasing ability to camouflage, all in a single body, still under evolutionary construction. This is the creature that rests in amber, a prototype, an experiment of nature in progress, inside the great laboratory that was the Cretaceous. The resins that gave rise to amber once covered the humid forests of Myanmar, protecting not only insects and leaves, but occasionally more complex beings. For a small reptile, a single misstep could mean both the end and eternity. When this creature was engulfed by sticky resin, its last movements were frozen in time. Every detail, from the scales to the contours of the eyes, was eternalized with almost surgical precision. Cretaceous lizard, Retinosaurus schkamtiensis. About 110 million years ago, a tiny creature crawled across damp leaves and intertwined roots, completely unaware that in a fateful instant, its story would cross through ages. Its name, Retinosaurus schkamtiensis, would be given much later by human hands. The resin, fluid and sticky, dripped from a wounded tree trunk, capturing the animal with almost surgical precision. In a matter of seconds, time stopped for that organism, and just like a three-dimensional photograph taken by nature itself, its body was encapsulated, sealed, preserved with a delicacy that science could never replicate. Every inch, scales, skin, muscles, and even internal structures was conserved with astonishing detail. Discovered in 2022 in the Khamti region of Myanmar, the amber fossil of Retinosaurus schkamtiensis surprised the scientific community not only because of its age, but also because of the perfection of its preservation. Unlike dry and fragmented skeletons, as is common in the fossil record, here was an almost complete specimen, so complete that it was possible to study the texture of its skin, the arrangement of its scales, and infer its lifestyle with precision. The preservation was so extraordinary that microscopic details were revealed: pigmentation patterns, dermal thickness, tiny claws, and even hints of original coloration. Retinosaurus is a fossil that seems to have escaped time itself, frozen at the peak of its life, with its eyes possibly still open at the very moment of its entrapment. With Retinosaurus came the opportunity to study an entire lizard, from snout to tail. Morphology revealed a slender body, agile limbs, and a scale pattern suggesting an arboreal lifestyle, likely feeding on insects and small arthropods. Among the most surprising discoveries was the arrangement of its scales. Unlike modern reptiles, retinosaurus displayed an almost ornamental pattern, with symmetrical rows and microstructures that suggested functions beyond simple protection, perhaps thermoregulation, camouflage, or even visual communication. This natural aesthetic, preserved for millions of years, is a direct window into the evolutionary design of that ancient era. Beetle from 99 million years ago. Trapped for 99 million years in a drop of resin, a tiny beetle remained hidden from the eyes of the world until it was revealed by modern science. The evidence it carries, microscopic grains of pollen firmly attached to its body, indicating one of the earliest chapters of insect pollination on Earth. Long before the appearance of modern flowers, known as angiosperms, there was another botanical landscape. Dominated by cycads, plants that resemble miniature palms with rigid leaves and discrete reproductive structures, this era saw the beginning of a revolutionary ecological relationship, the cooperation between plants and insects. And this beetle, preserved in Burmese amber, is the key that connects these two forms of life in a bond that continues to this very day. The discovery was made by Chen Yang Kai, paleontologist and fossil insect specialist, currently at the University of Bristol. When his supervisor, Diying Huang, presented him with the amber containing the beetle, the reaction was immediate. The enlarged mandibles with cavities covered in hair-like structures stood out to Kai as an unmistakable signature of a pollinator, not a predator, not a mere accidental insect, but a messenger of pollen, a traveler between flowers. With advanced polishing techniques and microscopy, scientists revealed details that would have been lost forever in a simple superficial observation. Within the crevices of the beetle's exoskeleton were clusters of fossilized pollen. Not just any pollen, but grains compatible with those produced by cycads. That detail alone reshapes part of the history of pollination on Earth. Preserved frog, Electrorana limoi. This creature, a tiny frog, could never have known that its fate would be to escape the ephemerality of time by being trapped in a drop of sticky resin. Its scientific name, Electrorana limoi, carries the weight of its history and the surprise of its discovery. Preserved in amber for 99 million years, this little frog is an almost intact window into the past, offering scientists a rare glimpse of a tropical ecosystem that coexisted with the last dinosaurs on the planet. Found in the amber of Myanmar, in Kachin State, it represents one of the oldest fossil records of amphibians in tropical environments, data of incalculable value to science. This paleontological jewel was discovered in 2018 by an international team of researchers led by Dr. Lida Xing from the China University of Geosciences in Beijing. The find was immediately hailed as miraculous, not only because of the frog's astonishing preservation, but also because of what it represents. Proof that frogs already inhabited humid tropical forests even before the cataclysmic mass extinction event at the end of the Cretaceous period. For decades, the fossil record of primitive amphibians had been scarce, fragmented and limited to only a few localities. Small animals with fragile bodies and forest-dwelling habits rarely fossilize. Moisture, predators, and time erase their traces almost completely. That is why the presence of 4 fossilized frog specimens in amber is not just surprising, it is a scientific gift. David Blackburn, herpetologist at the University of Florida, and one of the study's researchers, emphasized that the association between frogs and humid forests could be much older than once thought. Until then, this relationship seemed to be something more recent in amphibian evolutionary history. With Electrorana, that connection gains deep roots, extending back at least 100 million years into the past. Imagine for a moment the scene nearly 100 million years ago. A dense forest with a closed canopy where sunlight barely reaches the ground. Arachnids, insects, and primitive reptiles move among branches and leaves, while the first flowers timidly begin to bloom. Among this entanglement of life, small frogs leap in search of shelter and food. One of them, perhaps by accident, perhaps by curiosity, touches a sticky surface, the resin flowing from the trunk of a tree. Within seconds, its movements stop. Nature had sealed its fate.